Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by AGTC, Allergan and AbbVie Company, and Novartis. We'd also like to thank Accessible Media Inc., our national media partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
So before we talk about the specific grants that are being funded, can you talk us through a little bit about the process of how this comes together, how these grants are selected, and how we choose which ones are funded?
Oh, absolutely. So last year, as you mentioned, we held a research competition, and this was a research competition which was open to vision researchers across the country to put in their ideas for essentially whatever they thought was a great idea that they wanted to get a little bit of seed funding for. And so researchers could apply for up to $200,000, which is both a lot of money on one hand, but also in the scale of research can only be a fraction of some people's projects. And we then got them to put in applications. We assembled groups of experts to review the application. So this time we had two separate panels of experts, because not somebody who is an expert, say, for the front of the eye or the cornea won't be able to review grants that are about the retina. So make sure that we have the right people in the room. And they looked at the grants looking at a number of different items. So first of all, and sort of most importantly, what impact will it have for individuals who have eye disease? Is the question that the researcher answering, is it innovative? Is it impactful? Is it likely to move the field forward? And then they also look at some of the technical stuff. So what was the methodology? What were the experiments that the researchers suggested? If the experiments go as planned, are they likely to answer the question that the researcher is posing? And have they also identified risks that could happen, or challenges or barriers, and then identified potentially plan Bs or other things they could do if their first plan doesn't work? And then of course, are the experiments feasible? So, I mean, it's not easy, but you can think of like these amazing ideas, but if at the end of the day, with the money that we're giving a researcher, if they won't be able to pull off the experiments, it isn't necessarily a good use of money. So the researchers or the reviewers put in a lot of time reviewing all these grants. They get together, discuss every single grant. They make recommendations. Those recommendations go to our a board committee that makes sure that all the projects are within FBC's scope. And then it goes to the board for final approval. And we were so excited to be able to fund more grants than we expected. So 8 grants, as you mentioned.
Yeah, that's great. That's such a big number, 8 grants, and it's just really fantastic. So when we're looking at the scope of grants that are being funded this time, are there any sort of themes to these research projects, or are they all quite diverse?
I would say they're really diverse, and that part of it is the fact that we have expanded our mission. So now we ask for, people from all different types of vision research can apply. So not only retinal grants, but front of the eye. But it also reflects the fact that because we had some amazing donors last year who just gave us enough funding to fund more grants, we were able to fund a larger variety. So when I think about where the grants sit, I thought one way to think about it was which part of the eye and which diseases is the research going to impact. And so I put together a slide, which it's very simple. It's an image of an eye, and on the image, you can see the different parts of the eye. So working from sort of the left side, if you imagine the light sort of entering the eye, it has to enter the cornea first, which is a clear part of the eye, which protects the eye from damage and from environmental stress. And then the light continues to go through, it hits the iris, which is the black part of the eye, and that part of the eye opens and closes to let in different amounts of light. And then it passes through the lens, which essentially focuses the light towards the back of the eye. And then when it hits the back of the eye, that is the retina. And this is the part of the eye we do talk about a lot. And so the retina is a very complex piece of tissue, and I'll give a bit more detail later. But what it does is it essentially, it detects a light, and then it sends that light through the optic nerve, and that optic nerve goes to the brain where images are formed. So another part, if you sort of were looking at the back of the eye now on the right side of this image of the eye, a few other items I wanted to highlight were there are some retinal blood vessels which bring oxygen and nutrients to the eye. You have a part of the retina called the macula, and this is a part of the retina which is really important for detail and central vision. And then I've also noted where the optic nerve is, which, as I mentioned, brings signals to the brain. So if you can go to the next slide, Morgan. I have an image of the eye where I'm showing the retina blown up a bit. So you can see the complexity of the retina. And there are lots of different cell types. I'm not going to go into detail, don't worry about it, but the light essentially goes through the retina, hits a group of cells at the back called the photoreceptors. These are the light sensors. They create an electrical signal, essentially, a light signal, and they pass on the signal with the little processes on the sort of tops of their I was gonna say fingers, the tops of the cells. They make connections with other cells, and those electrical light signals pass from cell to cell to cell till they get to a group of cells called retinal ganglion cells. And retinal ganglion cells are the cells that make up the optic nerve. And then they gather all that light information and then transmit that information to the brain. So that's essentially how we get vision. And I think you can go to the next slide. I'm not actually sure what's on the next slide. Oh, this was the research competition. Yeah, so our researchers, 8 of them, their research spans all different parts of the eye. So if you can go to the next slide. I've just sort of broken down where some of the research fits. So in the cornea, the front of the eye, we have Dr. Stefan Ongtone and Dr. Ajitha Thanabalasuriar, who is with us today. We have for the retina, we have 4 different researchers, Dr. Rod Bremner, Dr. Anna Ells, who you're going to hear a pre-recorded session from, Dr. Julie Lefebvre, and Dr. Sakdev Sidhu. And then focusing on the optic nerve, we have Dr. Balwantri Chauhan and Dr. Michael Salman, who is here today as well. And this is obviously just some of the research we funded. So on the next slide, Morgan, I've listed all the other research that we are currently funding. So of course, we funded many more projects in the past, and this is a list here. I won't name all the names, but overall, we have 34 researchers and research projects that we are currently funding, which is a really great amount. You can see a lot of them, probably about 15 are focused on the retina, retinal blood vessels, and macula, but we do have projects spanning the entire range of the eye, the cornea, the iris, the optic nerve, and some that don't fit into these neat categories as well. So yeah, so that sort of, it was a long answer to your question about are there any themes, but I think that the main theme I think here is like cornea, retina, and optic nerve for this group of research grants.
I love this image and us talking about this because it just shows too how far FBC is coming too, because you know, if you'd asked us 5 years ago, it would have been all in the retina. So seeing everything that's branching out and looking at these other areas of the eyes is really exciting. So, you know, as you mentioned, we have 3 of our scientists here today or will be here or recorded here today to talk about their projects. And then over the coming months, we're hoping to have all of these 8 join us for a viewpoint to talk a little bit about their particular work. But just for today, before we talk to our researchers who are here, could you give us just a quick few sentences, I know it's hard, a few sentences about the 5 projects that we won't be discussing today?
Yeah, definitely. So I will start with the project that starts at the front of the eyes, Dr. Stefan Ongtone. As I mentioned, the other researcher is Ajitha, who will tell us about her own project in her own words, much better than I can later on. But Dr. Ongtone, he's at Sunnybrook Research Institute, and he's particularly interested in a corneal disease that's called Fuchs endothelial cell dystrophy. You might have heard of Stefan because he has won not 1, not 2, but now 3 awards from FBC in 2021, because he really is a really bright shining star. He also received a Clinician Scientist Emerging Leader Award earlier this year, as well as being one of the finalists on Eye on the Cure. In this project, he is trying to, like I mentioned, study this eye disease called Fuchs. So Fuchs is caused when there's damage to the corneal endothelial cells, and this leads to pain. It can lead to vision loss. It affects about 4% of the population over the age of 40 years old. So actually much more common than I personally knew. And currently having a cornea transplant is one of the only treatments available. So he's trying to look at other types of treatments. So what he's going to be doing is sort of taking a step back and trying to characterize what the different cell types are that are affected in Fuchs. So he's comparing cells from people who do not have Fuchs and cells from the cornea of individuals who do have Fuchs. Taking the cells, seeing if the cells are different in different regions of the cornea and how they differ between healthy cells and cells with Fuchs. And the hope is to use this information to develop more personalized therapies potentially in the future. And so moving on, I'm going to talk about Dr. Balwantri Chauhan. So he is studying glaucoma, which affects the optic nerve. And we're also going to talk to Dr. Michael Salman, who also is studying a disease of the optic nerve. But Dr. Chauhan, so he has developed a new technique to measure optic nerve damage. So retinal ganglion cells, as I mentioned, those are the cells that make up the optic nerve. And this is damaged in glaucoma. Glaucoma can be caused by many different, there are many different causes of glaucoma, but ultimately it leads to higher pressure in the eye, and this damages the retinal ganglion cells and causes vision loss. So right now, it isn't easy to measure how damaged the actual retinal ganglion cells are. You can measure if somebody is losing vision, but you can't actually measure how healthy those cells are. And so this is important both to understand the course of glaucoma, but also if you're developing new treatments and you want that treatment to cause the retinal ganglion cells to regenerate or to stop degenerating, there's no real way to measure this, easily. So Dr. Chauhan has developed a novel imaging technique which can take really detailed images of these retinal ganglion cells, and he's hoping to use it to identify biomarkers, and he hopes this will be a tool for other researchers to use as they're developing new therapies. And if you want to hear more about this project or more about glaucoma, April 12th, we have another viewpoint, and Dr. Chauhan will be joined by a clinician, Dr. Regine Kanji, who will be giving some more practical tips about taking glaucoma medications. So the last three researchers are all retinal researchers. And so the first project is Dr. Julie Lefebvre, who's at Children's Hospital, and she has a co-investigator on this project, Dr. Arjun Krishnaswamy, and he's at McGill University. And they're trying to understand how retinal circuits in the eye are impacted when photoreceptors are lost. So as I mentioned earlier on, the retina is made of all these different layers of cells. You have the photoreceptors, which are the light sensors. And in many different types of retinal diseases, like inherited retinal diseases, like retinitis pigmentosa, or age-related macular degeneration, photoreceptors die, and that leads to vision loss. What isn't as clear is what happens to all of those other cells in between. So the cells that the photoreceptors are passing signals to, are those cells still there? Are they healthy? Do they all have the right connections? And the reason it's important to understand this is if we want to develop restorative therapy, such as stem cell replacement, it's important that all these other cells are in place and can still transmit light signals. So for instance, if a new stem cell therapy was developed and you got new photoreceptors, but the other cells didn't have the right connections, they wouldn't be able to transmit those light signals properly. So Dr. Lefebvre and Dr. Krishnaswamy, they are using an optogenetics approach. This means they're going to be making cells that aren't light sensitive, and they're going to study how cells pass light signals essentially, and to see what happens when photoreceptors die, and if these circuits can be reactivated as well. And so another viewpoint plug, March 23rd, Dr. Lefebvre and Dr. Krishnaswamy will be joining us along with Dr. Brian Valios to talk about optogenetics and sort of restorative therapies. The next researcher might be familiar to many of you, Dr. Rod Bremner. He joined us last year for a viewpoint. He's identifying new drug targets for retinitis pigmentosa. So as I mentioned in RP and different types of IRDs, vision loss is caused by the death of light-sensing photoreceptors. And right now, there are gene replacement therapies that are in clinical trials, and they hold the promise of being able to stop cell death, but a new one has to be developed for every single gene mutation. So what Dr. Bremner is trying to do is trying to identify a more universal treatment. So try to identify proteins that cause photoreceptor cell death, with the hope that then he can use FDA-approved drugs, or drugs that are already approved, to block the function of these proteins and increase cell survival. So FBC actually funded this project in our last research competition. And then the reviewers thought that the progress that Dr. Bremner made was really impressive, and that this project deserved further funding. So we're really excited to be funding this for 2 more years. And our final project is Dr. Sakdev Sidhu at the University of Toronto. So Dr. Sidhu is studying, or has developed a new potential therapy for age-related macular degeneration and diabetic macular edema. So in both of these diseases, they're called vasculopathies, and these are diseases where the blood vessels in the eye, they grow out of control, and that can cause damage. So in these diseases, there's something called the blood retinal barrier, which prevents blood and other fluid leaking into the eye. So in vasculopathies like age-related macular degeneration, this blood retinal barrier is disrupted. This leads to fluid leakage, it leads to vision loss. There are treatments like anti-VEGF treatments, which many people have heard about, and they can reduce the growth of blood vessels, but they don't actually fix the blood retinal barrier if it's damaged. So Dr. Sidhu has developed a new treatment, and he is going to test if this treatment reduces disruption of the blood retinal barrier and improves vision loss in 2 animal models of AMD and diabetic retinopathy. And the hope is that this could be a new treatment for these diseases, either alone or potentially in combination with existing treatments. And final viewpoint to tell you about is on February 23rd, Dr. Sidhu won't be joining us, but one of his senior associates who is working on this project will be joining us to discuss this project. And we're also going to be joined by Dr. Bernie Hurley, who's going to talk about biosimilars, and Dr. Peter Curtis, who's going to talk about geographic atrophy. So sort of an AMD session on February 23rd.
Wow, that's wonderful. Thank you, Larissa. That's quite a lot. It's very exciting, and a lot of really different projects, which are really exciting to hear about. So I think we'll move on to actually talking to our researchers. So I think first I will play, unfortunately Dr. Anna Ells couldn't be here today, but I sat down with her earlier this week, and I will share, hopefully, a recording of that chat. Is that up on the screen? Yeah?
Yeah.
Okay, excellent.
It's Morgan and Morgan.
It's Morgan and Morgan,
yeah.
The Morgan and Morgan show. All right, hopefully we can hear this as well. Let's play.
Anna Ells, who's going to talk about her recently funded research grant here at FBC. Welcome so much, thank you so much, Dr. Ells, for being here, and welcome. Would you like to introduce yourself and tell us a little bit about your research to start?
Yes, thank you. Thank you so much for having me. And thank you so much to FBC for funding some of my research. I am a double trained in pediatric ophthalmology and in medical retina. And I practice medical retina and pediatric retina in Calgary, Alberta at the Calgary Retina Consultants. And I have studied, or my research focus in ROP has been almost my entire career since I was a fellow in pediatric ophthalmology at the Hospital for Sick Children in Toronto, and got interested in retinopathy of prematurity at that time, and continued to study the disease ever since.
Thank you. And can you start off by telling us a little bit about ROP? So what is retinopathy of prematurity? Who's at risk for this condition?
Yes. So ROP stands for retinopathy of prematurity. ROP is global. It affects infants throughout the world. And it affects, it's a disease of the retina, a vasoproliferative disease. So it affects the blood vessels in the retina, which is like the film of the camera in the back of the eye. And the infants that are affected are typically the smallest in terms of birth weight and youngest in terms of the gestational age in our populations. And it's been interesting. We've learned throughout the last 20, 25 years that not all infants in every country have the same risk profile. So infants that are born in high-income countries are really our smallest babies and our youngest babies. And infants that are at risk for ROP in middle and low-income countries have higher birth weights and higher gestational ages. So we've learned that in order to screen the babies and examine for the disease and make sure we know what's coming and how to treat it, we also have to know really which babies are at risk within each population. And a lot of work has been done on that as well.
What causes ROP? What's sort of the mechanism that causes that retinal change?
Yeah, that's a great question. So prematurity is kind of the they have to be born premature. And so what happens when in utero, as the eye is developing, blood vessels start to grow at about 12 to 13 weeks in utero. And slowly over time, the retina blood vessels make their way throughout the retina to the finish line. And the retina needs to be completely vascularized or full of blood vessels in order to function properly. So what happens if the baby is born or the infant is born at, let's say, 24 weeks instead of 40 weeks, the blood vessels are out of the gates, but they're only halfway to the finish line. And so what happens there is you have 50% or so of the retina that is without blood supply. But the retina still is very highly metabolically active. So that means the cells are working really, really hard at functioning, and can't do so without adequate oxygen and nutrients that comes from the blood supply. So then what happens is the retinal cells start to liberate kind of like a growth hormone, which stimulates the production of abnormal or bad blood vessels. And you think that, stimulating new blood vessels, that would be a good thing, but it's not a bad thing because these blood vessels are fragile. They grow in places that they're not supposed to be, and they break and they bleed and they scar and they pull the retina right off if nothing is done, and then the infant goes blind. So and it's a lifelong. Then the infant is it's a lifelong blindness that is mostly preventable throughout the world. It's not, we don't say it's 100% preventable, but probably 97, 98% of cases, depending on the country and economic status, are preventable. The blindness may be preventable. So
it sounds very similar to what you're describing to something like AMD or diabetic retinopathy, where all those blood vessels grow kind of uncontrolled and not where they're supposed to, and that's what causes the problem.
Yeah, exactly. It's in fact very, some of the biochemistry and stimulus for bad blood vessel production is really, really similar to diabetic retinopathy. Yeah, for sure.
Now, are there any current treatments for ROP? And also, a question I often hear is like, is there any sort of evidence for the long-term effects of these treatments on children? Because, so much of them, especially when they're premature, is still developing, still growing when they're out of the womb. Yeah.
So just a bit of history with respect to treatment. So we understood ROP existed in the 50s and 60s, but we really didn't start treating until late 70s, early 80s. And the first treatment was cryotherapy, so freezing the back of the eye and trying to really ablate or kill the retina that didn't have any blood vessels, what we call the avascular retina. And then in the 80s, we had better laser burning systems. And so laser photocoagulation, it's called, took over. And again, it was burning or ablating the peripheral retina that didn't have any blood vessels so that it couldn't produce this bad hormone or growth factor to stimulate abnormal blood vessel production. And so laser photocoagulation has been the mainstay of treatment, and about 10% about 60 to 70% of the babies that we screen get some level of ROP, but only 10% of those require treatment. And if you don't treat in a timely manner, things just continue to march on, and the infant can go blind. So timing is really, really important. So laser became really the gold standard and the mainstay of treatment until sort of the mid 2006 to 2010. And there's been a real paradigm shift in how we treat ROP to ablating or burning or killing 50% of the retina, to using a drug inside the eye that actually blocks the production of these abnormal blood vessels. And with time and with the right dose, perhaps may even facilitate revascularization, or the blood vessels continuing on their way to the completely vascularized the retina. But so these drugs are called anti-vascular endothelial growth factor drugs, anti-VEGF. We use them a lot in adults. They started with adults for most retinal vascular diseases, and then we slowly adopted them in infants. But with infants, this is a real special population of patients. So they're tiny eyeballs, tiny birth weight, and what happens is the drug, when it's injected into the eye, can be absorbed systemically or into the blood system, and these anti-growth factors that stop the blood vessels in the eye can stop developing organs in the premature infant. So we treat these babies when their brains are still developing, their lungs are still developing, their heart is still developing. So a lot of the research in the last 10 years has been focused on, okay, efficacy, meaning does the drug work? Yeah, it works. It's amazing. It's amazing. And we don't have to burn the retina. However, above all, do no harm. How much of this drug is staying in the eye? How long does it last in the eye? How much of this drug is getting into the bloodstream? And if it does get into the bloodstream, does it affect these babies as they become children? And that we have been looking at what we call neurodevelopmental outcomes to see over time if these drugs affect the infant's neurological development at age 2, at age 5 years. And that's been really, really challenging because prematurity itself is the single most important risk factor for neurodevelopmental delay. So how do you tease it out? Like, how do you know, okay, what's prematurity? What's the drug that we inject into the eye? What's the drug that they received for their bronchopulmonary dysplasia or their lung disease? So it's been really, really challenging, and many research teams throughout the world are looking at this. So the project that's been funded by FBC we've had prospective databases looking at these infants in the Calgary Health Zone since 1995. And with each project, we kind of add more to this database. And the database was an Excel spreadsheet. And, we would have part of the team go into the bottom of the hospital, collect all the data. So what we're going to do with our funding is to revamp this database, which has data from 1995. So looking at the different ways of treatment. So we started treating Calgary with drugs regularly in about 2015. So we'll be able to, we collected data on babies that were treated with laser and babies that were treated with drug. And we're looking at all the different characteristics of the eye and the lungs and the brain over time. And we're matching this chart data with imaging data that we have collected over time as well. And so this data set will be web-based, so it'll be really very, very secure. It's housed in the university clinical research unit at the University of Calgary, and it can be used by multiple investigators, and it can help contribute to the projects that we participate in internationally. So RP is such a small, it's a disease that doesn't affect, millions and millions and millions of people, like diabetic retinopathy and age-related macular degeneration. So because it is a disease with that affects a smaller population, we really work together around the world with our data sets to make sure that what can we learn? What can we learn together? What can we learn from each other?
Yeah, that's great. It's so interesting, and it's interesting too because, as you mentioned, like this condition can be preventable if it's caught early enough and if we understand enough about what's going on in the eye. So from what I understand, putting this database together, you're going to be able to look at treatments throughout the time and compare like outcomes and see which treatments are working, which may cause other side effects, that sort of thing. So how do you think all these findings could positively impact sort of the future of treatment for ROP?
So what we know is we know that laser works, and we know that these drugs work, and we know that the drugs in the short term are safe for the babies. We do know that, but what we don't know over time is whether or not the other systems will be affected. But as I say, that's really, really difficult to tease out, and it's going to take a lot of different kinds of data and around the world to look at this. But really that is the goal, is to not only compare the laser treatment, and we do almost no laser now. We do our primary treatment is intraocular injection at the bedside in the neonatal intensive care unit with these drugs. So we have looked, and other research groups have looked, and so we do know it's safe in the short term. We believe it is safe in the long term, but we're doing more pharmacokinetics, and what that means is that we're, also looking at what these drugs do in the blood system, in the bloodstream. But we're also looking at outcomes comparing different drugs, because different drugs are also used. There are 2 different drugs that are used. Well, actually, it's 4 drugs, but primarily 2 drugs that are used, and they look different. Their chemistry is a little bit different. How they behave in the eye is a little bit different. So not only are we looking at different major shifts in how we treat retinopathy of prematurity, but we're also looking at, okay, with this new treatment paradigm, how do these different drugs compare to each other? And some centers focus on one drug and some focus on another. So, we're going to be using some of this data set to compare. We use one drug, and in Toronto, University of Toronto, Sick Kids and Sunnybrook, they use a slightly different drug. So we'll be collaborating, comparing our short-term outcomes and long-term outcomes with this data set. So, we see this data set as just incredibly valuable to help us understand over time what are these outcomes, how do they compare to other Canadian centers, how do they compare to other high-income and middle-income countries throughout the world. So.
Yeah, that's fantastic. And maybe I'm showing my age, but I think of 1995 as not that long ago, but it's quite a span of time that we have all this data. So I think it's just going to be really interesting to see what comes of it, and hopefully, again, being able to compare those trends. Now, you did talk about how this project is funded or is focused in Calgary, and the records you have in Calgary. But then you mentioned some other hospitals like here in Toronto. Are there other registries or databases like this? Are there any chance of expanding to include the data from other sets, or is it just basically a working with other hospitals to understand their data as well?
Currently, this will be almost like a prototype for a potential ROP disease registry, and I think it just easy access, secure platform, web-based. Hopefully, we can expand this throughout Canada, so that when we do so much research together. But it's really cumbersome to data share, and it requires a lot of time, and it sometimes takes 6 to 12 months just to get agreements between universities. So if we can establish a national disease registry, which are really, really useful and add so much value, especially in these diseases that are less common, that is our goal.
Yeah, that's fantastic. And like you said, I mean, obviously, the Fighting Blindness Canada, our sort of origins are in retinitis pigmentosa, RP, and we do talk about a lot of sort of the smaller population diseases. So, we're together, we're many, right? So united together. It's always great to be able to get all that data together. And one thing I really have loved working with ophthalmologists is, and this community, vision science community too, is that everyone really wants to work together to come to solutions. So anything we can do to sort of remove those barriers is, I think, a really great step. Well, thank you so much for joining us today. I'm really looking forward to hear how it goes and how your project develops over the coming 2 years, I guess. And thanks again for being here. We really, really appreciate you sharing that with us today.
Oh, thank you. Thank you for having me, Morgan, and thank you so much to FBC for funding our work. It's really awesome. Thank you so much.
So that was Dr. Anna Ells in Calgary. We had a lovely chat, and her background there, I think she said that they have a place near Whistler, and it just looks absolutely lovely in the background. I just want to be there, actually. It looks so cozy. Okay, so that's great. So let's now see if Dr. Michael Salman is there. Yes. Hello, Dr. Salman. How are you?
I'm well, thank you. How are you?
I'm doing great. Thank you so much for joining us today. Could we start the same sort of question that I asked Dr. Ells at the beginning? Can you tell us a little bit about yourself and provide a bit of background on where you are in the country and what research you're doing?
Of course. So first of all, I want to thank FBC for funding me. It's a really great opportunity. A lot of funding is hard to get nowadays because, quite rightly, it's going to COVID. So basically, places like FBC has saved the rest of us that do the other thing that still needs to be done while there is this horrible pandemic. So I'm Michael Salman. I am a pediatric neurologist and neuro-ophthalmologist at Winnipeg Children's Hospital. I've moved there from Toronto just over 17 years ago to take on this position and never left. I started life as a pediatrician in training in the UK. And then went into pediatric neurology, then came to Toronto, was meant to be for 1 year in 1998, but I never left. In Toronto at Sick Kids, I did the rest of the pediatric neurology fellowship. And then I've always been interested in research in the cerebellum. That's the organ at the back of the brain responsible for coordination. So how come I moved to the front of the brain? Well, it was really just the way things sometimes work out. The cerebellum is a very hard organ to access. And there was a project at Sick Kids about children that have cerebellar malformation, and they were examining their eyes. And then I've always wanted to do more research and found an opportunity basically to record their eye movements so that I can study what's going on at the back of the brain, because that part is an important part that codes how the eyes move in these kids. So I got into eye movements, and I then went on to connect with Dr. Sharp. He is the late Dr. Sharp. He is a very prominent neuro-ophthalmologist in adults in Canada at Toronto Western Hospital. And he said, look, I'll help you record eye movements, but you need to do the fellowship. That'll give you unique training. No one has done that in pediatric neurology. It's marketable. And that, of course, had lots of extra training on the whole thing. And I was already at the end of training. But sure enough, I joined a PhD clinician scientist program, finished neuro-ophthalmology clinical fellowship and the research, and then moved to Manitoba. Now, I've seen the condition that I research like once in my residency training in the UK. And then I came to Manitoba and I kept on seeing quite a lot of it. So it turned out that we have the highest in the incidence, and as far as I can see in the world, a lot, and especially in the last last decade. So I got into it, and of course, as a neuro-ophthalmologist, I'm interested in, you know, everywhere along the visual pathways, front to back. So in this condition, the eye node is small, and some children have severe visual loss at both, meaning they can't see, and some are milder. So this is how I got into this research. I do neuro-ophthalmological research. This is the top and the biggest. And the other research I do is in cerebellar diseases. And the cerebellum codes a lot of eye movements. So there is a lot of overlap, as I found out.
Absolutely. So can you tell us a little bit more? I know you're looking at septo-optic dysplasia and optic nerve hypoplasia. So what are these two conditions and how are they different?
Okay, so optic nerve hypoplasia means the eye nerve that Larissa showed is small in size. And this happens congenitally, meaning during development in the uterus. It's small in size. It doesn't, the eye nerve does not have the 1.2 million axon, which are nerves, but there is less. And so the eye nerve is small, and that's what optic nerve hypoplasia means. The size is small, but at both it's small. Septo-optic dysplasia, you have a small eye nerve plus other parts of the brain can be affected. What are they? They can have a small pituitary gland, or a gland which is in the middle of the brain called pituitary. It secretes a lot of hormones essential for survival, and they have a non-functioning or partially functioning or poorly formed pituitary gland. It can also affect how the brain itself is developing. So if there is those other things in the brain, whether it's the pituitary or the other parts of the brain affected as well, we call them septo-optic dysplasia. These are all congenital, meaning you're born with it.
Are there particular risk factors for these conditions, or are they quite random? How does that happen?
So in terms of risk factors, first of all, we don't really know why this condition happened. There is only a small percentage of patients. They talk about 1, 2, 3%. There is a genetic cause. Despite extensive investigation, the rest we don't know. So so a lot is unknown. So I am looking at my project at risk factors. So risk factors have been known about and talked about for decades, but these all come from doctors that have seen 1 or 2, or a group that have seen 10, 20, 30 patients. There are larger groups. There is one well-known, for example, in Los Angeles, that have a database, just like your prior researcher. They established a database across the country in the US. And all the kids, if they get sent, besides the ones in California, they've investigated those. So these are where the risk factors come from. Small reports, small number of patients reports, where they've linked different things: drugs used during pregnancy, medications, exposures to smoking, alcohol, drugs during pregnancy to the unborn fetus. All these have been discussed. There's also research from Sweden that have shown that young maternal age, and the same as well in California, young maternal age and the first pregnancy is at risk. There is more evidence for that. They've done a larger study and compared it to existing national averages. So yes, there is tons of risk factors. Like there is numerous toxin exposures, medications, and maternal demographic, young maternal age, and first pregnancy. So those are the risk factors.
How are you going to be studying these risk factors in your project?
So I'm doing it differently. It's really the next step, because it's not good enough to have a few patients and say they all share one thing. Their mothers were, I don't know, young or smoked during pregnancy, because it could be smoking is prevalent in that particular city or population, and it could be prevalent in moms that do not have kids with this condition. So you need to compare it to something else. So the studies, the big studies in Sweden and Los Angeles, well, California, I should say, which also is other parts of the US that have been referred, they've compared it to national averages. So they have the statistics data, and they said, well, the national average for smokers in pregnancy is 10%, but the mothers we have is 20% of them smoke. So then they said, oh, smoking must be important because they're more prevalent in the mothers that have these babies. What I'm doing it is different. We're lucky here in Manitoba. We have something called the Manitoba Center for Health Policy, and they have for decades collected data, medical, educational, social, hospitalization, and coded them, and they're all anonymous. So what I'm doing is I already know who has this condition here. We've worked very hard in the prior years identifying those cases. So what we want is to comparing them with people, with pregnant women that lived in the same area, have the same type of demographics. So we're matching them by age, by sex, by where they live, because that takes care of a lot of things like the income, etc. So geographically, we're trying to match them. So my cohort of patients will be matched by 5 as many controls. So each person that have this disease will be matched with 5 people, same age, same gender, that do not have the disease. And then I'm gonna look backwards in time and see the mothers of those controls. I'm gonna check and see, what did they do during pregnancy? Did they get exposed to smoking, alcohol, drugs? How old were they when they had their kid? What number kid has the disease? Is it the first child, like some studies said? Is it another, like the second child? So I'm basically comparing the children that have this condition and their mother characteristic during pregnancy to 5 as many controls. That makes the study stronger, and then comparing their characteristics when the mothers were pregnant. That is a higher scientific value. Everything has value. It's the next thing, because here we are in 2022. Genetics doesn't explain a lot, and there are numerous risk factors. Which one is really the ones that matter? And I'm doing something else, which also I'm really fortunate to be able, because I never thought about it. We could also match the children that have this disease with their siblings that do not have the disease. And then we're asking the question, did their mothers, during the time they produced the siblings without the disease, smoke? Did they take alcohol? How old were they, etc.? So that's how the design is different.
It's very interesting. I like that idea. It's more like comparing apples to apples, right, versus apples to oranges. You're really kind of trying to narrow in on what the differences are. So just lastly, what do you hope that the impact of this research will be for people who have SOD or ONH?
So we have a high incidence of this disease, and we want to know what's going on in Manitoba. And the question is this really the same? Because I've had discussions with other people that are interested in this disease, other researchers, people that deal with this. And I'm told in the prairies, not just Manitoba, it is high, but here was the first place that it was quantified, not by me, but I'm working to the next step with this project, where they found it's really high in numbers in Manitoba. So the question is this across Canada? Is this and not just Manitoba? And also importantly, is this really more common and we're not recognizing it around the world? So we don't know why there is such high numbers, number one. We don't know why the majority of them have the disease in the first place. So high incidence and high concentration in Manitoba would be the impotence for me to find out. If we can identify what risk factors and then see if some of them are modifiable, like smoking. I already presented in the proposal the preliminary data for smoking. It turned out smoking is a big deal. It really seems to be way more commoner in mothers of kids with this disease than mothers in kids without the disease. And if we can say, we know smoking is bad, and we know smoking in pregnancy is bad, but we can then say, look, the developing optic nerve is particularly vulnerable, and we can decrease the incidence. It will also give us insights. Is there a toxin? How is it impacting the development of the optic nerve? Is there something in the smoke that on the uterus, inside the fetus, that affects somehow a chemical from the smoking gets into the uterus, fetus, brain, optic nerve, and affects it?
That's really interesting, and I'm looking forward to learning more as you go through your project. So I think I'm going to pass it over to Larissa now, but we'll come back, Dr. Salman, at the end if there's any questions for the audience as well from the audience.
Thank you very much.
Over to you, Larissa and Dr. Thanabalasuriar.
Thank you. Thank you, Dr. Salman, and thank you, Morgan, for both of those interviews. They were really, really interesting. I've read a lot of the proposals as the sort of applications we're going through, but it's really nice to hear from the researchers explain it. It's much easier, to be honest, than reading some of the applications. So welcome, Dr. Thanabalasuriar. You said you prefer to be called Ajitha, so I'll just call you Ajitha if you don't mind.
Yes, please. Go ahead. Call me Ajitha.
Yeah. So Ajitha is an assistant professor at McGill University. So the same as with the other two researchers. Can you just introduce yourself briefly and give us a little bit of a background into how you came to be doing this research?
Thank you, Larissa. And thank you to FBC for having me today and funding my research and taking interest in my research topic. So just changing base actually quite a bit from the other two interviews. I am actually a basic researcher. I'm an assistant professor at McGill University. And I just started in August 2020. And my research really focuses on the front of the eye, on the cornea. And my general research theme of my whole lab is focused on immunity, particularly innate immunity and infection. And I'm really interested in the interface between this and host pathogen interactions. And so what I sort of brought to the table for my research is this novel technique to understand host pathogen interactions called intervital microscopy. And using this technique, I can actually image and visualize disease progression. And during my postdoc, which I did actually in Calgary at the University of Calgary with Dr. Paul Koobs, I developed a way to image the cornea of a mouse using a mouse model and understand the progression of keratitis, so an inflammation of your eye, which I think is going to be your next question, but I won't ruin it. Spoiler alert. But yeah, so basically I've used this technique, intervital microscopy, to understand mostly bacterial infections, and particularly the cells, innate immune cells called neutrophils, which are actually circulating throughout your body on a daily basis, protecting us from infections, and how things can go right and things can go wrong on the surface of the eye.
Okay, that's great. I already have all these questions about immunity because I know it's a really popular topic and a topic I find really fascinating, but I'll stick to the questions for now, and we'll see if we've got time afterwards. So you mentioned that for this project, you're studying something called bacterial keratitis. I was wondering if you could just tell us a little bit more about what that eye disease is and who potentially is at risk of bacterial keratitis.
Okay. So bacterial keratitis is essentially an infection of the cornea. And this is actually an interesting disease because you could be completely healthy and develop it. And often it's just, chalked down to bad luck. And this always scares my audience because one of the main individuals or susceptible populations for bacterial keratitis are actually contact lens wearers. And usually when I poll my audience, about 30 to 50% of them are wearing contacts at that moment. And so that's one of the major risk groups are contact lens wearers. And I'll talk a little bit about why they're a risk group. But other populations that are, at risk are people who are immune compromised. People who've just undergone surgery are quite susceptible to keratitis as well. And this is mainly because they're immune compromised. So their immune system in their eye is not working as it's supposed to. So why are contact lens wearers susceptible to infections? It's mostly because we're taking on and off our contact lenses on a regular basis. And this can cause small abrasions on your eye. And there's bacteria everywhere. You can't run away from microbes. They're on, our skin, they're on our fingers, they're in water, everything. And so just by bad luck, if you happen to get some of those bacteria in your eye, they can cause an infection and they can cause inflammation in your eye, making it hard to see.
This was not one of the questions we suggested we'd ask you, but I'm going to ask it anyways. I was wondering, so is there anything that a contact lens user can do to reduce their risk of keratitis, or is it just, as you're saying, just purely bad luck?
Yeah, so all the new, with contact lens wearers, all their solutions and things like that are quite sterile, and they recommend that you take them off daily and clean your contact lenses. Don't use tap water to clean them, things like that. Those are all procedures to prevent that. But again, one of my students, he was like, once I accidentally used the wrong solution, I burnt my cornea. These accidents do happen, and if in that context you're just around something that has more bacteria than it would normally have, or it wasn't a table that wasn't clean and you touch your eye, you can get an infection. So it's just really bad luck because we can't escape the microbes around us.
So if somebody does get an infection, what sort of treatments would they have? So
basically, the treatment right now, as it stands, is antibiotic eye drops. And if it gets worse, if your keratitis continues to progress, and it doesn't get your eyes don't clear up, and it's quite red and inflamed, then you have to go. Eventually, you end up in the hospital with intravenous antibiotics. And if that doesn't fail, there's really nothing else out there at the moment to help stop the infection.
So if the infection can't be stopped, does that impact somebody's vision, or does it just lead to pain? What can be the impact of that?
Oh, absolutely. It can result in vision loss and severe pain and basically blindness at the end.
Okay, so extremely serious. So even though there are treatments out there, mm-hmm. There's still a potential. So I know that is sort of the purpose of your project, trying to understand the relationship between the bacteria and the immune cells in the eye. So I was wondering, maybe you can just give us a little bit of a primer about how the body fights off an infection in the eye normally.
So our eyes are actually really phenomenal organs. Originally, they were thought they were immune privileged, essentially saying that they didn't have an immune system. And now we're starting to see that they actually have this really intricate immune system that sort of functions in like this stealth mode, clearing and protecting our eyes. And so starting off with, our tear film has tons of antimicrobial compounds, including immunoglobulins and enzymes that can break down bacteria if they end up on the surface of your eye. And then the structure of your cornea is covered in corneal epithelial cells. And these corneal epithelial cells are actually really great at protecting our eye. They are able to take in any bacteria and actually kill them. And they're this tight sort of seal that protects the rest of our eye. And again, it's when these protections are lost is when you often have infections, right? Again, for example, when you're taking on and off your contact lenses or during trauma. And actually, right now, the research in immunity, especially in the eye, is really flourishing. And we're starting to see that they're a resident population of immune cells that are in between those corneal epithelial cells, like macrophages that, you know, observe the milieu of your eye for infections and take them up before without causing too much inflammation. And recently, in the last couple of years, there was a paper that came out that actually when you're sleeping, say something gets into your eye and you go to bed, it's not really bothering you. During sleep, cells called neutrophils can actually come into your eye and clean up debris and clean up bacteria on the surface of your eye. And you know, when you wake up and you have like, my daughter calls them eye boogies, but those are actually those neutrophils being released from the surface of your eye after they've done their duties while you were during the closed eye time. So our eye actually has a lot of different immune systems and different functions that can protect us, but it's when those things go awry that infections can come.
That is fascinating. I had no idea that that was what like that crust on your eye was. So it's it's just like the sort of the housekeeping functions at work throughout the night.
Yeah.
Huh. Another reason I guess you need a good 8 hours of sleep every night, right? To like not only for the rest of your body, but for your eye health as well.
Exactly.
So you mentioned that you might not be able to clear, like you give somebody antibiotics, but some people aren't able to clear it. Is there a reason why somebody wouldn't be able to clear the infection, or why some infections become antibiotic resistant?
Yeah. So there's two reasons why you can have an antibiotic resistant infection. One is naturally, you know, bacteria are everywhere. They're trying to survive in their niche, and they can pick up antibiotic resistant genes. So for every antibiotic, there's going to be an antibiotic resistant bacteria, and that's just how it is in the environment. Some of them are very hard to find, like in the Arctic, and some of them are just in the soil, and, you know, again, bad luck. But what I found with my research is that in an attempt, when all your other eye defenses are broken and you have this infection that's growing, in an attempt to clear that infection, you recruit these cells called neutrophils from your bloodstream and other parts of your body, and they move onto your cornea. And when these cells move in, their main function is to, you know, stop that bacteria from getting deeper into your eye. They're not thinking about antibiotic resistance at the time, and they form these things called neutrophil extracellular traps, which are really great at protecting your eye from this bacteria moving in, but they also completely destroy your corneal function. And then they actually encourage the bacteria to form into this structure called a biofilm that's super antibiotic resistant. So these biofilm structures can be made by a bacteria that's not resistant to the antibiotics. So it's actually susceptible. But once it forms into this structure, it becomes resistant. So in an attempt, our body is trying to protect our eyes, actually form these like super bugs on the surface of our eye, which actually leads to antibiotic resistance. And so really my research is to sort of help our neutrophils, help these immune cells not do that, you know, protect our eye without encouraging this ultra transformer bacteria that become antibiotic resistant. And so it's sort of the yin and yang of our immune system. You know, their function is to protect us, but in some senses, we lose the use of that organ. And so my research goal is really to target these cells, these neutrophils that are sort of our first and last line against these bacteria, to change their function in order to really clear the infection without promoting antibiotic resistance.
So interesting. So I wonder if you can tell us, like, maybe in a sort of a simple way about what exactly you will be doing in the next like 2 years to try to like meet this goal.
So in the next 2 years, one, I'm going to be visualizing the function and the behavior of these neutrophils using my intervital microscopy, and then understanding why they're forming into these neutrophil extracellular traps. Because neutrophils actually have many ways that they can kill bacteria. They could take it up, like those corneal epithelial cells, and kill them inside their body. They can form these degranulations that can sort of release enzymes to slowly kill the bacteria. But for whatever reason, they're forming these traps in order to sort of trap the bacteria, but encouraging them to grow into these antibiotic resistant biofilms. And so we're going to use different techniques such as imaging and RNA sequencing to define these neutrophils and what's going on with them, and then potentially find genes that we could target to stop them from doing one function neutrophil extracellular traps, but increase their ability to, for example, phagocytose and kill the bacteria inside the body of the neutrophil.
So like ultimately, how, like I'm thinking like sort of long term, I know it won't be at the end of 2 years. How do you think this research will help patients? Could it lead to the development of a new drug, or what do you think, how would this be able to impact?
Yeah, so really what I want, well, obviously everybody wants to stop blindness, but really, you know, when you have a patient who has this antibiotic resistant infection, I want another line, something else that we can give them, something else to help to sort of save their eyesight. And this would be it, you know, whether it's in combination with that antibiotic. Once you start to see that they're not responding to antibiotics, we give them this alternative therapy to promote that. So to sort of encourage the antibiotics to work, or alone as a single therapy to help prevent that the eye from becoming completely opaque and being able to at least see in the future. So really that's, to stop the last line being blindness. Can we keep their eyesight?
My last question is, I was actually just wondering, because you mentioned that the neutrophils prevent the neutrophil extracellular trap.
Yeah.
They prevent the bacteria from going further into the eye. So what would happen if the bacteria does go further into the eye?
Yeah, so at this point you have complete vision loss. And it's actually a little bit scary. So if you don't have those neutrophil extracellular traps or the neutrophils functioning perfectly, in our models, we find that it actually can, the bacteria, depending on the bacteria that's infecting your eye, some of them are quite motile, and they can actually move into the back of your eye, into your retina, and sometimes hijack your optic nerve and move into your brain. And that's what we saw in, you know, in our isolated models. But yeah, so in the absence of these traps, it can actually cause quite severe infections to the back of your eye, so you lose your whole eye. But so that's really what those neutrophils are protecting you from. But can we change those functions to keep that protective role, but prevent the blindness?
That makes sense. So they are doing a really, really important job, but just how they're doing it, maybe try to change that a bit.
Exactly, yeah.
Well, thank you. I'm going to actually bring Morgan and Dr. Salman back. I really enjoyed talking to you, Ajitha. Thank you for explaining your research to us.
Oh, thank you.
Great, thank you so much. I'm going to give our speakers just a moment to take a breath, and we'll let everyone sort of ponder that. If you have any questions, please feel free to put those into the Q&A box. And if that is not accessible to you, then you can always also send an email to education@fightingblindness.ca. And I'm just going to ask my colleague, Jamie, to join us for just a moment. Let's see if we can get Jamie in here. And Jamie's just going to talk to us for just a couple moments about some ways that you and the audience can get involved to help support this important research.
Hi, can you hear me? Great. Thank you so much, Morgan, and thank you to the speakers. This has been really interesting. It always is. I love the Viewpoint webinars. They're amazing, so thank you. My name is Jamie Alexanderson. I am the Senior Manager of Development at Fighting Blindness Canada. I understand there are over 80 people registered for this webinar today. Morgan, that's fantastic. Thank you all for joining us. I'm just going to take a few moments of your time. I'd like to talk with you about how you can get more involved with Fighting Blindness Canada to support vision research, some of which you are learning about here today, which is incredible, and it would not be possible without donor support. So these are ways you can try and think about getting involved. I know that some of our listeners now are already wonderful donors to Fighting Blindness Canada, so thank you for your continued support. But here are some other ways maybe you'd like to get involved. Our events are a great way to get involved. Participating in Cycle for Sight, joining us at Comedy from the Couch or Ride for Sight. And I think coming up, we actually have Screens Off for Sight on Saturday, March 19th, which is a wonderful event. I know many of us will participate in that. And of course, if you're thinking outside the box, right? So if you're thinking about marking a milestone or celebrating a special occasion, perhaps consider making a gift in honor of a friend or a loved one or a colleague. We have lovely cards on our website to choose from. Another way is, visiting our website. We talk about advocacy and public awareness, making your voice heard on issues that matter to you. And if you've ever thought about supporting vision research for generations to come, we have more information on our website on how to make a special gift in your will, or learn about the tax benefits of making a stock donation. And another way that we've started promoting a program, an employee matching program. I didn't know this, but did you know that many employers will match an employee's donation to the charity of their choice? So there's so many creative ways and inspiring ways to be able to support Fighting Blindness Canada that are meaningful, that can bring meaning to you. And so please visit our website at fightingblindness.ca or give us a call. I know Morgan, our host, will be sending out an email after this webinar with all of our contact information. We look forward to hearing from you. So enjoy the rest of the webinar and the rest of your day, and bye for now.
Thanks, Jamie. Great. Let's see here. All right, so we'll go to some questions. Now, let's see here. Okay, so let's see what I have here. So I think the first question I have here is for Dr. Salman. So why do you think that the rate of SOD or ONH is so high or on the rise in Manitoba? Is part of the reason that doctors are, more aware of it and they're becoming better at diagnosing it, or is there something else going on there?
Thanks for the question. This is why I'm doing this research. We really don't know. There was an epidemiological study. An epidemiological study tell us about increasing incidence, how much there is of a certain disease that came out on some of the patients that I'm investigating by colleagues of mine before I got into this. And they found between 2009 to 2014, and comparing that to the following 5 years, it jumped. And it could be partly better recognition, but it's the same, it's us the same. And I don't know where we can recognize it anymore, us doing this work. I think it's because there is something else going on. In that epidemiological research, usually like in the UK, it said it's 10 in 100,000. In Europe, they did a multi-center. I'm sure there is a lot of missed cases. They only found 99. I have at least 111, but actually there is more because I've only focused on Manitoba, but we also look after Northwestern Ontario and Nunavut. So I haven't even included them. So we can make it epidemiological and just talk about Manitoba. So in that work published in 2017, they showed more in northern communities. So northern communities, there will be small towns, reserves, and there is more of this condition. And the question, but it can happen to anybody else in Winnipeg. It really can happen to any pregnant woman, especially if they are younger than 24, it seems, and first pregnancy. So this is it. I don't know if we have higher exposures to toxic substances than the national average in Canada, but I'll certainly be looking at that. So if I find, say, smoking among mothers that have kids with this disease compared to mothers in Manitoba without kids of this disease, and then say compare it to the national average of how many mothers smoke during pregnancy, that would be a way to say it. But it doesn't seem to stick to one population. It could be anybody, Caucasians, First Nations, Inuit, Mennonite, Hutterites, whatever there is. It affects everybody. But as I said, it could be we have the northern community, according to that study done 4 years ago, five years ago published, shows it's more common in northern communities. The question is, do they have different risk factors? Is there more? We'll sort some of this out with this. So nobody knows. And I'll be interested to know if other prairie provinces have it. And also importantly, Toronto, Ontario, Quebec, the Atlantic region, we have only one children's hospital, and this condition, you can't really just go to a family doctor. This is complicated. You require, you know, these kids, I didn't tell you, they have also, some of them, if they have the septo-optic dysplasia, seizures, developmental delay, the gland doesn't work, they need replacement, otherwise they could just collapse and die. These hormones from that gland are essential for survival. So I don't know if it's just Manitoba. It's just we seem to see a lot of it. We're all aware of it. I don't think it's just us diagnose it quicker than anyone else. It is recognized by ophthalmologists and neurologists. I suspect there is toxic exposure, substances, something, medications, and it might be higher in Manitoba or prairie provinces.
Yeah, well, it sounds like there's a lot of possible factors. So it'll be interesting to see what comes of the research. Great, a question for Ajitha now about biofilms. So can these biofilms that you talk about, can they form in other diseases or in other organs or parts of the body, or are they specific to the eye?
Thank you, Morgan. That's a really good question. And yes, they can form in other parts of your body. And many diseases that we are dealing with are caused by biofilms. And one in particular, if you think about cystic fibrosis patients, so these are individuals who have a genetic mutation resulting in them having poor lung function. They can actually develop bacterial infections in their lungs that form into biofilms. And these can become antibiotic resistant and quite life-threatening in the case of the lung. So yes, so understanding the mechanisms of how this is happening in the eye can actually help in many different diseases.
And another question for you, what happens if the bacteria go further into your eye?
Yeah, so Larissa alluded to this a little bit, and it's not a happy result. So of course I use, a rodent model to look at all this, and in humans, this usually doesn't happen because we are treating them and there's surgical interventions. But in the model, when we have, in our rodent model, when neutrophils are not forming those nets, neutrophil extracellular traps, the bacteria can actually move further into your eye and onto your optic nerve. And we actually found them in the brain. And when this happened, the mice, the rodents are still alive and they're not able to see, but they start to have cognitive dysfunctions. They're not able to walk straight. They're starting to, weigh onto one side, and their faces become enlarged and inflamed. So yes, it's not a happy ending.
Not sound pleasant. And it's interesting because that's what's so fascinating about the eye too, is it really is just an extension of our brain. So it's not surprising that it kind of travels back that pathway if it goes untreated.
Exactly.
Okay, so I think I have one last question. This is for both of you. So the question was, if your research is successful, what would your next steps be? So maybe we could start with Dr. Salman.
So I think the next step, say if I find an exposure, a toxic substance, smoking, alcohol, drugs that would cause it. So one option would be to educate people. So I live in a children's hospital, basically, where we encounter a lot of people. There is, a method to educate whoever comes, and there are projects done by pediatric residents about advocacy, and we can dissuade people even more from smoking or taking alcohol. So that's one possible option, but I think smoking seems to already come up on the pilot study, and what I want to do next is convince one of our researchers who's a basic scientist. Otherwise, I'm going to run to Ajitha to get her to do it. What I want to do is, if it's smoking, then I want to know, there's lots of substances in smoke. So I want to know, has anyone found a toxic effect of a substance within the cigarette on the developing optic nerve? And even to also, so I'm talking about developmental things. Well, what about after people are born and they have the full normal optic nerve, if people get exposed to that, what does it do to the already developed optic nerve? Is there any implications for older, like after birth exposure, that could affect the eye in a certain way that we do not know? But perhaps getting into an animal model to see afterwards if we can identify something from the literature and see if we can prove it or disprove it in a mouse model, for example. Whatever model is available that simulate the humans would be the one to do. There is already somebody, but I want to see what it is. I want to have the backup from this study before I go and kind of collaborate. So that would be one thing, basically. It's really the modifiable risk factors that I want to prove, and then I want to prove by this study, and then take it to the lab, and then take it back to humans to see if we could make a difference, if we have the backup scientifically that this is a huge deal in causing this disease.
Great, thank you. The same question for you, Ajitha. What would be your next steps after this research if it proves to be successful?
Yeah, I would, first of all, I would love to collaborate with you, Dr. Salman. So anytime you have any questions for a mouse model, please give me an email. And second, yeah, so first, I would like to find a targetable gene in neutrophils to stop those extracellular traps and enhance other types of phagocytic killing. And so one of the things I did not talk a little bit about was I did have some experience between my postdoc and my faculty position in a pharmaceutical company. And ideally, you know, I would partner with them and we would develop a therapy that can target these neutrophils, whether it's monoclonal based therapies or it's a small inhibitor. So really put this into a drug. And then sort of, I would probably be talking to Dr. Salman to do human studies and look at this in patients. So, you know, we would be sort of crossing roads and he would be moving into the mice while I'd be moving into humans in the future.
I love that collaboration, networking at Viewpoints. Very good. All right, well, thank you both so much for being here today and answering all of our questions and just being generous with your time. It's really been wonderful to hear from you and excited about hearing from you again, you know, at the end of the year, next year, to see how these projects are shaping up. So if you enjoyed today's webinar, please do visit our Viewpoint page. Larissa mentioned in her talk early on about some of the different viewpoints that we have coming up. Next month, we will be recognizing AMD Awareness Month and talking about AMD. I have a friend who's just joined me. School is over. And then we will be doing optogenetics in March, and April we will be focusing on glaucoma. Please just take a moment to fill out that survey that pops up at the end. It really helps us as we're planning our future webinars. And thanks again for joining us. Please stay safe, stay healthy, and I look forward to connecting with everyone again very soon. Have a great day, everyone.
Thank you again.
Bye. Thank you.
Thank you.
Thank you so much again for having me.
Same. Thank you.